that wraps up the elections. That's a major change of the guard here. But our mission is the same. So, okay, if there isn't anything else, let's see who's the best to introduce our wonderful speaker. I know John Windsor knows a lot about this fella. Probably not there.
He's there. He's just got his mute. Yeah, yes, John.
Can I put you on the spot, John?
Yeah, put me on the spot a little bit, but a couple of years ago, I was responsible for contacting Dewey Caron to give us a lecture on swarming, and our communications got all messed up, and I panicked, and I started making a PowerPoint thing, and I had to finish it the next morning because that was the day of the meeting, and I get this email from Dewey. He goes, "What the hell's going on? When am I supposed to speak? And fortunately, he just carried on like he knew what was going on. He's a professional, consummate professional, retired PhD from Baltimore?
University of Delaware, close.
University of Delaware. Okay. You can't pick up a honeybee textbook or article practically that he doesn't have his fingers all over that stuff. He's just the go-to guy. And so he's been really super generous for our club. He's spoken to us several times, and I'm really pleased that he's back tonight. So Dewey, take it away. You're going to do a great job. Everybody's looking forward to hearing what you have to say, and we never leave disappointed.
Well, thank you so much, John. Thank you for that introduction. I do, in part of that capacity, is I do serve also as on the board of the California Master Beekeeper Program. Excellent program. Just doing a superb job out of Davis with that program. So I applaud all those members that are part of the California Master Beekeeper Program. If you're not, check it out. I think you might be very pleasantly surprised about how it can offer something more to your beekeeping. In our Oregon program, we started back about 2012 with an Oregon Master Beekeeper Program, and it has really made a difference in terms of the level of what the clubs are able to do. A lot more sharing of clubs, which is useful, sharing of speakers. So I really heartily recommend the California program. I know some of you may have also participated in the one out of Montana, Jerry Brobenshakes, or the one out of Cornell, or perhaps are a member of one of the others. But you're homegrown. The California program is just superb.
If I can interject.
Go ahead, John.
Quick, we are kicking off our California Master Beekeeping Program. Humboldt County, this coming Sunday night, is our first review session. I think we have about 10 people signed up this year, which is the biggest we've ever had. So we're going to be calling on you guys that are already apprentices to help us out, I think. But anybody out there that's even considering it, get a hold of Janine or myself and come on board. We'd love to have you. And that's all I'll say.
Yeah, those kitchen table sessions are extremely, extremely valuable. I think, I applaud the group for having a local effort to try to work with that. It's a bit daunting, most of us have been out of school for a very, very long time, and the idea of tests and doing this and having to jump through these hoops is a bit of a stretch, but you'll be richly rewarded for it. All right, going to talk about integrated pest management. I'm going to talk in more general terms, but talking with Eric earlier, some of you may have very specific questions. So as I go along and I don't get to them, let's make sure we get those very specific questions answered. Let's start with a definition. IPM, Integrated Pest Management, is a strategy that helps utilize multiple techniques to fit the context of each situation, with a long-term goal of reducing or preventing pests In almost all cases, we can't get rid of them, but we can, in a number of cases, control them. But there are very few where it's a one and done, one shot, or the magic bullet that will get rid of them. So that's the concept of integration, of not using just a single tool, but trying to develop the tools that will work together and be most effective. In the real world, the ideal world, it involves a threshold to establish treatment guidelines. And much of the bees where we don't, and I'll talk a little bit tonight about how kind of with varroa mite, for example, our major pest, the threshold we have may not be very useful right now, this time of the year. It may not be serving what we need to have it do. Okay. So once you kind of get your handle around what the pest is, what it's doing, what its life cycle is, and all of those aspects, then trying to establish, do I need to treat it, or can it go for another look, or will the bee county solve its own issues without my intervention? All of those. So treatments then, but when we decide we think we need one, or our threshold says that they're over a certain level, we probably better do one, are selected and applied to minimize risk to ourselves, our own human health. And we're in some cases using a pesticide, a chemical designed to kill. And we can harm our own health with that. And using the pest's own enemies, the beneficial organisms that might be harmful to the pest, but also might be very helpful, maybe not directly in a beehive, but in the environment of the beehive. So beneficial or non-target organisms. In other words, we want to focus, let's say, on killing varroa mites. But if we do some aspect to something else that's in the colony that there's a lot of other mites serving other purposes, for example, if we're also killing those other mites, what's we may not understand the other purpose of those mites. So those we'd call non-targets. The varroa mite is the exception. There are probably in your hive right now 40 different mites, and 2 may be very harmful. The varroa mite, and if you have an infestation with tracheomite, those are 2 of the mites that are harmful. But the other 38 have a role to play, and they're not harming at all. In fact, some of them are either benign, not doing anything, or are actually being very helpful. And of course, then the wider world, our bees are part of the environment that we share with them. And of course, they share with lots of other organisms. So minimize risk to us, our own, the applicator, ourself, our own health, the other things that are out there, the beneficials, non-target organisms, and the environment itself. Okay. So we're going to look at different methods, different types of means of trying to do that treatment. And kind of the concept that we often lose sight of in treatment of a pest is that we should try to start with the less invasive treatments, those that are more benign to everything, to our own health, to the environment. And of course, our human nature is that's not what we do. We go for the atomic bomb right away. We go for the pesticide, kill it with a chemical right away. Chemicals actually should be on the hind end of our thinking of, well, what am I going to do to help my bees with this particular pest situation? Not on the beginning of that list, but they should be on the hind end of that particular list. So that's a little bit what we can think of as visualizing IPM. In the big picture, there's no silver bullets when it comes to managing pests. Effective management involves many layers that are often work together to reduce, that have to work together, or work together to reduce populations, to have thresholds that are sustainable. Okay, so that's sort of where I'm coming from with a concept in terms of pests. And we use this, and you'll see this as I go throughout, that our pest control triangle so that we're looking in terms of aspects that we might do to prevent a potential pest from becoming pestiferous, all the way up to intervention, meaning we're doing something to kill the pest. And as we get into the chemicals aspects, there is an increasing level of toxicity as we go from some levels of chemicals to what we call the hard chemicals, often the synthetics. It doesn't have to be synthetic, but in other words, made in the lab. But you know, some of our natural ones we have enhanced, and they are just as toxic as anything that chemists can make up in the lab. So this is our sort of our triangle, and we'll walk up that triangle. Now, as you know, honeybees have many stressors. The 4 Ps, pests or parasites, pathogens, poor nutrition, and pesticides. I like to add a 5th, and that is people, you and I, the beekeepers, as well as the non-educated bee public. And I'm going to focus, of course, on this aspect, the pathogens and pests tonight. Okay. So what are some of the pests that we might look at? Topping all of our list are going to be varroa mites, and I'm going to come back to that a lot tonight. They are external parasites, but they are much more than that. They're external parasites on really hyped up on steroids, is in what we're going to talk about. Yellow jackets and wasps, certainly. Ants can be a pest. Again, you know, it may not be as significant, but among some of the ants, they can be more serious pests than others. Spiders and predators outside, you know, everything from anything that can eat other insects, basically, can be a predator. Bugs, certainly, beetles, some of the others as well. There's a group of animals other than the tiny ones, mice, skunks, and bears. We're certainly very familiar with bears. It's the type of pest where you've got to do the prevention. You've got to build the bear fence before the bear comes and starts discovering what great riches are in that bee colony. Birds, there are a number of those that will eat bees. And again, it's okay if one is eaten, but if that one happens to be a queen on a mating flight, that is a serious issue for the colony. Potentially a condition is going to lead to a laying worker because there may not be able to replace the queen. Zombie fly, which we know as an internal parasite, the maggot gets into the bee, and as it is coming out in its life cycle, the fly's life cycle, when it changes from the maggot to the chrysalis stage, its pupal stage, it comes out through a weak spot, and that happens between the thorax and the head, and so they decapitate their hosts. And that's not good for survival, of course, of the hosts. They're in many colonies, but not a serious pest under most conditions. Some serious insect pests, of course, are small hide beetles and wax moth. There's another mite I've mentioned already, tracheal mites, and for some of us, probably one of the most serious pests are humans. Here, pack colonies in the winter, the tipped over, stuff scattered about, and then colonies exposed, certainly at the wrong time of the year, that will be poor for their survival. So it's sometimes difficult. One of our tasks as beekeepers is to be that intervention, is this a pest that has a potential to become serious, or is this, and will it remain a pest that is a minor pest? And what you might individually consider as a serious pest for you. So every time you go into your colony, there are ants, the sugar ants on top of the colony. And so they're on you, and of course ants will bite and sting, and so that you might get bit and stung and get welts from that. So if it's every time you go in your colony, you might consider ants a more serious pest than than someone that may or may not see any ants on the top of their colonies, or if they are, they brush them off and open up the space so that the bees can get in there, and the bees police their own space and they get rid of their ant problem. So minor to perhaps more serious pests. And that's our own value judgment, of course, is this something that is a pest that I've got to do something about, or is it something that the bees are going to handle, and I've got to maybe just give them a hand, like lifting that cover up just a bit so there's more space, or reducing the amount of equipment on the colony so that the ants then don't have a place to start their rearing of their young, because bees are policing in under the inner cover. And the pathogens, and again, right at the top of the list is varroa mite. When we get our southern collapse of the winter kill colonies, maybe Israeli acute paralysis virus, IAPV, or maybe acute bee paralysis viruses, one of those two perhaps. Talking with Eric of perhaps some people seeing a little bit more of the second one of the paralysis viruses this spring. Some characteristics to look at. We do know that that varroa mite is not just simply a predator on the adult and the growing pupal stage of the bee, but it's also a organism that helps certain viral compounds in the bee that exist naturally in the bee to become very prominent. And these include the IAPV, ABVV, and especially the DWV, the deformed wing virus. Whether I mean, bees have a lot of viruses. We have a lot of viruses. In most cases, we're okay. We get a viral infection, and we'll then have our cold, or maybe something more serious, such as we've gone through with this pandemic, a lot more serious. But the same happens with bees. So bees too are going through pandemics of their sort, and it's because these viruses, these particular viruses, all of a sudden become dominant in the colony, overpowering other viruses, replicating, multiplying within the bees. And when they reach epidemic proportions, boom, the colony will collapse. We'll see this as bee colonies dying in the fall, after we put them to bed, often in November, December, or the colony, when we go for our first inspection come spring, it's a dead out. It just doesn't get through the winter period. Sac brood is a virus that is in both larvae and adults, and we see a characteristic shape of what it looks like as a larva, but adults with sac brood don't seem to have any outward signs that we can diagnose what's going on. And when sac brood becomes more prevalent in some colonies, then it's gone from this status as a minor pest to perhaps an actionable item, an actionable pest that maybe we have to do something about. Other pathogens, bacteria, fungi, certainly in bees. The bacteria ones there's a lot of bacteria in us and in bees all the time. There are, again, many of them are benign or very, very beneficial. We can't live without the bacteria. Same too with bees. Most of them are not harmful, but there are a couple, and we've labeled those American foul brood and European foul brood. They're both bacteria. One is a killer. The AFB kills colonies. EFB weakens colonies. Hardly ever will kill a colony, but it is possible. And again, the intervention is different. And so our role as a beekeeper is if we see something that might be a bacterial infection that's going on, and of course we're looking for the signs of it in our bees, then we need to make a decision. Is this a colony that is suffering from sac brood? Is it because the brood are not becoming into the pupa stage? Is it something that is European foul brood? Is it chalk brood of the fungus? Is it American foul brood? if they get to the cap stage, but no further than that. And again, our aspects of actionable items will vary with each of those, and also with the severity, except for the killer, except for American foul brood. That's an actionable situation like varroa mites at all time. What we need to determine is what's the level of the infestation. For example, in a colony, you can find in your bees, you can likely find the American foul brood in a percentage of your colonies. If you go and look for the pathogen, in other words, look for the bacteria. If you've ordered bees in as nukes, if you've ordered bees as queens and replaced your queen with someone else's queen, you bought a queen, package bees, nukes, you're going to find a percentage of those have the bacteria. What is not the case sometimes is that we see the sign, we actually see the bacteria, what it's doing to the bees itself. So too with varroa mites, we'll find all colonies with the varroa mites. We can't really easily measure the levels of virus, but we've got to assume if there are varroa mites, there's a chance that they're going to enhance one of these very serious viral infections. The fungus, the chalk brood and stone brood, and finally in the adult, a very special type of fungus. It's been classified as very number of things as an adult, but it no longer, of course, is called. We're no longer going to be calling it nosema. We're going to be calling it something morpha, poromorpha, or something of that nature. The genus name has been switched, not the species name, or is in the process of doing so. So the adult, this would be an example of American foul brood. I should have brought that in when I had that. And this is European foul brood, and its symptom we're looking for here is European foul brood. Is we're looking for larvae that are out of place, twisted, in the wrong color. And the characteristic in the middle shot of American foul brood is that before the larva or the prepupa completely dehydrates, it is gooey and sticky and it ropes out. So this is what is called a ropey test for American foul brood. Should have brought those in a little bit earlier. So the colony is a complex organism. And the sum is equal to more than just the individual parts. So we often will label it a supraorganism. It accomplishes things that one individual alone would not be able to do. And they share those activities, both spatially and through time, through the season. So in our looking at our bees, our diagnosis of do we have something that might be an actionable item? Do we have a pest that can get out of hand or is out of hand? We can look really at kind of in three different ways. We can look at the population of adults in the colony. Populations that when others are gaining in numbers, it's declining or it's staying even. Small populations as a superorganism will have more difficulty defending against those that are larger, for example of being robbed, and are harder to treat for mites. And yet, on the other hand, a smaller population can grow fast and grow out of mites. So it can grow out of a mite problem, or keep ahead of a mite problem, at least for a while. So the adult hive population, brood pattern, looking at individual larvae for signs of disease. The spotty or shotgun pattern can signal disease, doesn't have to. And our job as the beekeepers to figure out what is it saying? What does that spottiness say to us? What's going on? Why is it spotty? Looking for that solid brood coverage, all brood roughly in one area of the same age. And then, of course, if once we develop the ability to see eggs, that looking and making sure is one egg per cell, Rarely we'll see two, but then more than one can signal some other issues that are going on. Our looking at our hive actually starts outside, so external hive characteristics. We can simply look at the entrance activity, get an idea if they still have brood, if pollen is coming and going, but there could be reasons why pollen is not available to the bee, so it's not by itself an indication that there's something that is different. Many of us will use the debris board, the screen board insert, and once you get skilled, if you have one, if you have that modified bottom board, I hardly recommend you get used to looking at it, because it can give you those early warning signs of something is amiss in your colony. So gaining the skill of looking at that insert, that debris board underneath the colony, to try to pick some of these things up. And then mites themselves, I'm going to keep coming back to mites, because that is the genesis of at least 70% of any problems we might have in a bee colony throughout a year on average. And so what we want to know is not, does my colony have mites, but we should be able to come up with some sort of an estimate of how many mites does my colony have. Again, our treatment strategy is not to get rid of every last mite that might be in the colony, but to try to manage that. And that's where this pest management strategy will come into use. As I say, it would be great if we had a very simple test to test thresholds. Well, we do, and that is to take a sample of adults and check for how many mites are on the adults. But there is problems with that sampling this time of year. This is very useful later in the year, but right now, this time of year, that's not going to be adequate. I want to come back to that point in a minute, in a little bit here. So looking at the population, here's a brood frame, big, nice pattern, solid pattern. Very few cells that do not have a brood at the same stage of development. Those show up as empty cells, nothing in the cells. Queen could have laid an egg in it, or may have missed the cell. Real solid pattern, but unless we have brushed bees off or shaken bees off that frame, a part of our assessment is the population of adults. Is that high population adequate to take care of that brood? And there could be a number of reasons why at the time we're doing our looking at our bee colony inspection, there could be a reason why at that point it's not adequate. So we're always thinking and treating it. We should be at least periodically, and at least a kind of a minimum twice a year, treating the beehive as a crime scene, a CSI, where the detectives looking at and seeing what might be or could be an issue. We ought to twice a year look at that. That versus this brood pattern below, number again of adults, cells that are open that should not be. In other words, once these cells are capped over by the bees, then there usually is no reason to uncap them, unless something is not right. And the main thing why our bees are doing this uncapping, you can see that here in this lower middle portion of the cell. I don't know if you can see my pointer going around those. There are a number of cells that are uncapped. In other words, the capping is no longer intact on top of the colony. And those are bees, we can roughly call them hygienic bees. Those are bees that, for whatever reason, have detected a mite. We think because the mite is reproducing, the baby mites, and they're looking for it. So they may uncap a whole bunch of cells looking for what cell has that mite in, to run the reproducing the adult mite out of the cell. Well, in the process of doing so, they have stopped development of these pupa that are in the cells, unless they recap the cells. And the genes for recapping are entirely different from hygienic bees that have this ability to detect the mites, to be able to smell the mites, and then go do the searching. So we need both types of genes in our bees, the alleles that not only have them look for the mites, but then make an assessment of what's in the cell itself for developing sister, and if it still is healthy, to recap that cell. But that's again a different set of genetic capability of our bees themselves, not just being hygienic, but also hygienic being doing the finding, but then salvaging some of their young instead of removing all of them. I mentioned the bacterial diseases, so I have this slide with American foul brood from Michael Wilson. We look for this most serious of the brood diseases in the cap stage. So we are looking for cells that are sunken. This capping is not that little bit dome-shaped capping that look greasy, that have tiny perforations in them. And if there still is something that's not completely dehydrated, taking a stick or a toothpick, opening up the capping, the rest of the capping, looking at the color. The color is going to be this cafe au lait or caramel colored, and then seeing if it goos out or ropes out. We call our ropey test. The other aspect that we need to also look at as well is on the lower rim have the dehydrated prepupa, larva and prepupa, have they melded into the lower cell level? So we're looking here, we've got the top bar at our belly looking towards the bottom bar. So we're looking at the bottom of the cells, and we can see these scales. We call these scales. And each of these scales has encapsulated bacteria, thousands, millions of them in most cases. And it will remain as that scale until the bee lays an egg in that particular cell. And once there's living material in the cell, then the spore can come out of that spore stage into the active vegetative stage. So here, this spore, the spore broke on this case when an egg was laid in there, infected the very young larva. The bacteria grew out of the gut into the body cells, and then all throughout the body, it reproduced at the expense of the bee. And by the time we found it, it was this gray, caramel colored, dehydrating remains of what could have been a replacement sister. And the overall pattern here you see is very much of a spotty pattern. It's simply because the eggs that are being put in these cells are not leading to a capped stage. Now, not every one has the spores, the bacteria in the cells. So there are among here healthy. There are healthy capped brood, but there are likely others unhealthy. We picked out this cell, but up here it looks like there's a very much of a sunken grayish looking cell, greasy looking cell. There's one that's perhaps up here. And again, here's this close up of these spores. So it's like an extra rim, and this is only on the bottom of the cell. So I've used this as an illustration that to be very effective in managing a pest, or in this case, the pathogen, the bacterial pathogen, to be very effective, we got to know about it. We got to know what signs, what signals to look for. All right. So we're doing like an autopsy for dead organisms, a necropsy of our dead colonies. And if we're looking at a colony that has died out, this is this scale. There's not going to be any living material left. It's going to dehydrate. So the scale is the stage we look for. If it's a organism that is still going, the colony is still going, we want to look and find these cells that are at the stage of breaking down by things such as a ropey test, or by this sunken capping, this sunken perforated capping. We got to look. We got to go that extra step to try to confirm. Well, this is a case of, I suspect, of American foul brood. All right. In this case, what we're looking at, American foul brood would be our suspect. And so then I've got to do something. The colony is not going to cure itself of American foul brood. On the contrary, if this was a case of European foul brood, other signs and signals, then we might ask the question, well, how many cells are being affected? What's happening to the brood production of a colony that has an active infestation of European foul brood? And should we intervene? And then we've got some treatments that we can do for European foul brood. So now we go through our decision tree of okay, what are we going to use? Now, in case of pathogens, we're not talking of pesticides usually. We're talking of an antibiotic. So if we have a European foul brood, the easy solution is to think of the antibiotic that will kill the bacteria. And of course, that is the antibiotic terramycin, which will do that. But there are other interventions that we might do. And of course, that we're going to have to make that decision. Is this actionable? Is this case of European foul brood in this colony or in my apiary so severe that I need to do something? And if I need to do something, do I need to do it now, immediately, without closing the colony up? Or can I close the colony up and think of something that I can manage this on a long term? And of course, with the case of European foul brood, the bacteria is there. It's going to be there next year. It's going to be there in the fall. So our decision is how are we going to help the bees get through this potential bacterial reinfection and breaking down of larvae later in the year or the next year? And one of our answers could be to replace the queen, to get different genetic stock in there, that maybe we have bees that are not as efficient at policing themselves, so policing the bacteria. So there's a whole series of levels of decisions that we might make. And so that's all within this realm of IPM. We're going to use this pest as our example. It is both a pest and also this spreading of the pathogen. It's not really the pest that's the issue. It's the spreading of the viral pathogen. It's enhancement of these particular ones that cause the epidemics, the DWV and the acute bee paralysis virus is the Israeli acute paralysis virus. But as with a lot of things, we do not have an antiviral agent, antibiotic to work with against the virus. So what do? We're going to try to attack the vector, in this case, the varroa mite, enhancing those viruses. So it's really not the varroa mite that we're concerned about. It's the varroa mite having the possibility of transmitting the viral pathogens, enhancing those that cause some real problems. Here's a list of some of those vector diseases by mites. I mentioned the acute bee paralysis virus as one of them. This has the appearance of bees being shiny or greasy, like they've been wetted perhaps. The bees with acute bee paralysis viruses are trembling, sort of the DVAC maneuver, but they're not asking help with grooming from a sister. They're just sitting there and sort of trembling and shaking. They are very poor flyers for whatever reason. We may see them outside of the colony on nice forage days, having trouble taking to the air. They'll come out, they'll climb grass stems, they'll climb anything in the vicinity, climb on a beehive itself, but then just not have the ability to gain flight at that particular point. So a number of somewhat subtle characteristics for the acute bee paralysis virus. There's a second bee paralysis, and that's chronic. Some of the same characteristics, but the difference is acute is able to reach these epidemic levels and kill an entire colony. The chronic is over a longer period of time. You don't see nearly as many bees infected with it at any one time. And whether it ever reaches levels to kill the colony is debatable, but certainly the acute bee paralysis virus does, and it will kill a colony pretty quickly. DWV, the deformed wing virus, and this is an illustration of the bee that's facing towards the, if it's a clock face, towards 12, facing upward, in other words. You can see a bit of this greasy appearance to her body. She doesn't have the same body hairs of her two sisters forming the V below her, but particularly the wings. The wings have never expanded when she first emerged from her cell, which is a very short period of time that this must occur within the very few minutes that when she emerges from her cell. That the wings have to expand. Well, she's not able to do so. And it's because her body is ridden with all of these virus particles that are affecting a lot of things inside of her body. It'll affect her lifespan. Most of the viruses do that, but also their ability for memory, their ability to efficiently perform tasks, to do the learning that they must do to perfect tasks, etc. And of course, with DWV, their ability to fly. They will live for a little while. This bee will live a little while in its colony, but then depending on how many bees are infested with the virus, how many are still healthy, the healthy bees are going to have to remove the body of this bee, take her out, detect it and take her out. And so if you've got a heavy infestation of DWV, and a lot of bees being born without the wing expansion, the colony quickly goes into a collapse from the epidemic of the virus itself. IAPV, the Israeli bee paralysis virus, implicated in CCD, along perhaps with the APV, the acute bee paralysis virus, as well as perhaps nosema disease of the adults. There seems to be association with IAPV with nosema disease. We've never solved what was the issue with CCD. We still get those symptoms in a colony of a colony collapse. And now, though in most cases we are equating them with DWV or ABVV from the virus transmitted by the varroa mites. Bees that don't close their wings, a little bit of example of the two bees that are the V below. The wings in the should be, unless we've we put smoke into the colony or breathed on the bees or whatever, those wings should fold over the abdomen of the bee out of the way, so they don't get rubbed against other bees, don't get rubbed against comb and wear away. If they don't, we'll call this K-wing, particularly when the front wing and the hind wing, if you look at their appearance, look like a K. And these two bees, the wings are not really folded well, but K-wing is their inability for virus particles that are in the mites, are in the muscles of the thorax, affecting the ability of the muscles to fold those wings, a very tiny muscle, but a critical one, to fold the wings one on top of the other, on top of the bee's body when they're not using them. The other thing that happens is the wings that hold them for whatever reason are not functioning correctly and are rasping against the wings, and they just aren't able to fold their wings normally. We equate this with tracheal mites rather than varroa mites, but we also see this with this DWV characteristic as well. I'm sorry, with the acute bee paralysis viruses as well, the not being able to fold the wings. And I guess this makes the point as well that many of these don't have. We shouldn't be looking for the magic sign, the magic signal that, aha, this is what is going on in this colony. We kind of do a look at a number of things, the number of adults, the brood pattern, certainly, the health of the brood in some cases, the health of the adults, looking for these signs such as K-wing or deformed wings. Not one thing will necessarily tell us what is going on in the colony. and we can use as general characteristics. Our colony is not doing as well as it should, as are some of the others right now. They're not having a good amount of brood. There's not enough adults to cover the brood that they do have when nights become cold, or their cluster is going to form off of the brood or not enclose all of the brood area. Bees out front that are not acting as they should, coming and going, or doing orientation flights, for example. Not paying attention to a queen, not getting good queen retinues. All of these are general characteristics that something is wrong. And then we've got to do the CSI. We've got to figure out from looking at these signs and signals that the bees are giving us, the unhealthy of the bees. Not all are going to be unhealthy. The unhealthiest, what might be the case. I don't know if any of you have seen black queen cell virus, another one. The developing queen simply stops development and becomes black. It's not brown or yellowish. It's distinctly black. So all of these, in one way or the other, are mite vector diseases. And so why we have no treatment as such, no antibiotics for the viruses themselves. So we go after the vector. We go after the mite as a way of trying to help the bees help themselves. All righty. As I indicated, knowing our enemy, knowing what our enemy is and what kind of life it lives and why it might be in a bee colony and what damage it might do to a bee colony if it has any numbers, is part of our issue with determining, the playing field of the enemy. So in our varroa mite cycle, what is important to understand is that the varroa have two phases in the bee colony. The phase when they're on the adult bee body. Here's a bee just emerging with a foundress mite and a sister mite emerging from her cell. So this was not detected by the worker bees. So she went through pupal development with the mite feeding on the pupa. And that is called a phoretic or dispersal phase. Phoretic means just hitching a ride, and that's not actually correct because they actually feed on the adults. And then the reproductive phase. In the reproductive phase, as that larva goes through about its fifth molt, in other words, they grow, they double their size every day. That's the way they grow after the eggs, the first 3 days, develop into the larva. At that last stage where the larva is filling the interior of the cell, a foundress mite, a female, drops off of a host, the honeybee, worker, a nurse bee, for example, and taking care of feeding of the young, and then goes and hides in the brood food at the bottom of the cell for the day or so that the pre-pupa spins its cocoon. It goes around and around spinning its cocoon, and worker bees on the exterior surface put a capping over it. Following that, she is actually maturing eggs that she is able to mature in her body because she is fed on her host in that phoretic stage. And roughly in the first couple of days, she can lay an egg, and that becomes the first egg as a egg that'll develop in a male. And then roughly every day thereafter, she can lay another egg, and that'll be a female. So she then opens a feeding hole on the pupa. She puts her fecal matter on the cell wall to signify where that opening is, where she has opened up a feeding hole. And then as her young go through their development, she and they come to that same feeding hole and feed on the developing pupa. If it's just one mite, the bees may not detect the developing bees, but if there's more than one mite in the cell, bees are usually pretty good. If there's any hygienicity in the adults here, these nurse age bees that are walking over the cappings, they can detect the developing mite family inside of the cell, and we'll open up the cell. So that's that behavior we call hygienic. But if that doesn't occur, then the mite is able to successfully reproduce herself if she's entered a worker cell. If she does this whole development in a drone cell, she's able to reproduce herself more than once, developing into as many as 3 adult females, mature females, whereas in a worker, it's roughly just over 1, on average, 1 mite per cell that she's able to enter if it's a worker bee. And then when the bee emerges, out she comes, the foundress, the original mite that went in there, and a mature daughter, a mature female. They will then find a suitable host to feed on the fat body, not the bees that are leaving the colony generally, but the bees that are still within the brood area. And then as they then feed, they then wander over around to the thorax of the bee, and at a suitable cell, they're able to detect this larval development stage, just like the bees do. They're able to detect the right stage, and then they drop off of the host, the bee they're riding around on, to restart their own cycle. Most mites can go through 5 or 6 mite cycles. The average is fairly small, low. It's only about 3 life cycles for any one single mite that she's able to do this. In other words, reproduce herself within a cell. Now, this happens in our European bees. This mite is native on bees that are native to Asia. Other species of apis, other honey bees. And in those other honeybees, this doesn't happen. The mite is not able, the varroa mite is not able to reproduce herself in a worker bee. She's only able to reproduce in the drones. And so the Asian bees that have this mite, Varroa destructor, and other Varroa species, are able to control this mite because they control how many drones they rear and when they rear their drones in a season. Unlike the European bees, they don't continue rearing drones for much of an active season. It's a very short season which they could rear their drones, so that the drones are available for reproduction to provide the sperm, but are not year round. They're not being reared year round in colonies, although the temperature would allow that to do so. So what we have in terms of a pest, and this is often the case with a pest, is it has found a host that is particularly suitable for it, the pest, to be able to go through its reproduction and go through successfully through its reproduction, and then have a fairly large population because it's able to do that reproduction over a period of time. It transferred to the European honeybees when European bees were brought to parts of Asia and then brought back to other parts of the world. And with it, the Varroa, the pest came, but not the pest that will control Varroa, or the host, in this case, the European honeybee, having a biology that simply is very conducive for mites to be able to grow and reproduce for a good part of the season, not just for a very limited time in the season on its host. Let me start with sampling as our principle. Perhaps at this point I can ask for some questions before I talk a little bit about sampling. Are there, if you want to
Anybody have any questions? Go ahead and shout them
I
out.
have a question. What is it? The black queen, when she hatches, she cannot mate, correct? I mean, she's not viable.
Not viable, correct? Right. Yeah.
Because I had one, and she was worthless. I mean,
Yep. Yep. Indeed.
She can't mate, correct?
Correct. Right.
I have a question. Can you confirm, did you say that Nosema is going to be renamed?
Yes.
Okay.
The genus name.
Oh, okay. And
We're still can call it Nosema disease,
okay.
but the pathogen will have a different, its name will not be Nosema.
Okay. Thank you for clarifying that.
Sure. I think the same thing, like we still call mites on adult bees as phoretic. It's not the correct term, but we're still using that term phoretic. Others? By the way, a new article in California Agriculture proposes yet another name for what we know as the Africanized honeybee. It's probably about the 50th name that has been suggested to rename that particular ecotype of honeybee, the so-called Africanized honeybee. Not
They're
so-called, that's
calling
what
it
we call
calling it less than friendly.
What's the proposed name?
Scutellata, Scutellata genetic, Scutellata. We're no longer like to use the word term race either for our bees. Scutellata, that its first word is Scutellata something. I'll have to look that up. Maybe I can get back to you right away. Do you get the California Agriculture? If you get the California Agriculture, the article by the UC San Diego people on Natives Lab, a very good article on Africanized bees in California. Good summary. All righty. Let me try to go on. Mite sampling. Monitoring mite loads can save hives. We want to monitor at least twice a season. In fact, many of us like to begin in April and then continue on until we put the bees to bed in whatever month that might be, end of October or November. Some like to do twice a month. I'm going to talk a little bit about how now in April and May and into June, our method of monitoring is not adequate. It's not going to work. And then we want to then try to come up with a number. So not that we're looking at, does my colony have mites? We're not looking for just to find the mites, but we're looking at trying to make an assessment of how many mites the colony might have. So typically we use either the sticky board, a passive method. So there at the top tip of the finger is some mites on a sticky board or our debris board. Lots of other stuff on there. So you're looking at that. And the assessment is, gee, I hardly see any. And so I'm going to continue monitoring. Gee, I see a few. Here's one, here's another one, here's another one, but there's not many. It's been in a day and I'm seeing less than 1 an hour. Some people would say 12 or fewer, or 6 or fewer, 1 every other hour. And so I'm okay. I'm okay with my colony and the number of mites. I'm going to assess again within 2 weeks or within a month. Or the third assessment is, God, darn and all, there are a lot of mites here. It's beyond my counting ability. So, counting not every mite, but an assessment of how many mites are there. It's a bit passive because the mites have to fall to the sticky board. If you are an aficionado and use a spacer at the bottom, you like to use the bottom, an extra bottom bar, the one that has the slatted rack, what we call slatted rack. The mites will fall onto the rack rather falling than on a sticky board. If you have a homemade one, you may have used a very tight mesh on the screening material. And so the mites fall on the screen, but then not through to your sticky board. If you have ant predation on your colony, they're going to remove the mites. So this is as a sampling method to try to determine one of those 3 levels. Very few, enough that I've got to start worrying, or doggone, I got to do something immediately. A very passive method of looking at that determining a threshold. So then you will use something, a sugar shake, an ether roll, which no longer is in favor, alcohol wash. These are jar methods. So here we have a large mason jar, and we've used the sugar shake, a powdered sugar. through a screen top. We have to get every bee thoroughly coated with sugar, so there's no spot on their body that doesn't have sugar. This interferes with the mites, and then the mites fall off, and then we're going to shake out the extra sugar along with any mites that come out. Or an alcohol wash, we can substitute non-sudsing sugar, soapy water. People like the Dawn Ultra that doesn't sud as much, where we shake this liquid onto our bees. In this case, we've got a shaker that's joined in the middle with the screening in the middle. We pour and we put the bees in our top jar. We just pour our liquid on there. Shake. Oh, actually, we don't shake. We swirl. We want to swirl this. We shouldn't be shaking this. Swirl this, and then try to get the fluid to settle right on through all of those bees, and then in the liquid portion on the bottom, look at the number of mites. So many of us will buy this from Vito Pharma, the Easy Check. That's basically a white basket in a plastic container, easily removable lid. We put our sample of bees, 200 or 300, and it's the basket is marked as to the number. So you fill it up to the 200 line or the 300 line. Then add the liquid over the top of that, swirl that for at least a minute. Or if you have access, shake them for about 10 minutes in a shaker, a moving tray, for example. And then what we're going to do is we're going to look at the liquid that's in the bottom. So here we're taking our sample. We like to take the sample from the brood area where we maximize the possibility of finding mites on the nurse bees. So we make sure we don't have a queen. We shake the bees that are there adhering into some sort of a tub, in this case, a white dish pan. We look again. We looked on a frame for the queen. We look again in the tub to make sure we don't have a queen in there. Then we're going to take a half cup, an ordinary kitchen measure, a half cup, scoop out, shake the bees into the corner and scoop out a level half cup of bees and put that into our device, into the basket that's going to go into our device. Add the alcohol, 75, rubbing alcohol is fine. The 92% is better. Leave the better hooch for yourself because it takes a while to do this. So if you're going to use vodka, save the vodka for yourself and buy some alcohol that's for the bees. And then after about a good minute of swirling, you look at the bottom. So here's our basket, the white basket holding in this case, a sample of 300 bees. A half a cup is roughly 300 bees. And then we're looking in the liquid that's through, so that we've washed these bees of the mites. The alcohol causes the mites to separate quickly from the bee's body. And then by swirling, they separate from the 300 conglomerate of bees and get into the liquid below the basket on our measuring device. With the powdered sugar, unfortunately, there's more errors because the bees can get caught in the powdered sugar. The powdered sugar is going to clump unless you really thoroughly coat all the bees and let the bees then dehydrate in the sun. And you really need to shake, and it's shaking for at least a minute, dehydrating for in full sun 2 to 3 more minutes. In a cloudy day, it doesn't work quite as well to have them dehydrate in the sugar, and then shaking again for another minute. So it's a lot longer to do an accurate sample. So the alcohol or substituting the non-sudsing soap is a little bit faster method. And so then, depending on the time of year, if we're doing a 300, the half cup of bee sample, using a jar method, either the sugar shake or the alcohol, we then can come up with an idea for a threshold. And we're going to end up looking at a percent infestation. So we're looking at the number of mites. We're divide the number of mites by the number of bees that we're looking on. So if we have the top line of that Vito Pharma mite check, that's the 300 sample. So we divide by 300, and then we're going to make it a percentage. So we then multiply by 100. So if you have in that sample 3 mites, then we would have a 1% level of infestation. So 3 per 300 adult bees is 1 per 100 or 1% infestation. So then on time of the year, March to May, we can call that roughly spring. If we have anything that's more than a 1% level of infestation, we should then make a decision that our mites have a possibility of killing our colony. And if we want to do some intervention at that time, then we're going to think of a treatment. In the summer, there's more bees. And in the summer, the mites are more likely to be on worker bees. Summer here defined as the hot months, June through August, peak population for the bees. So we will often use a higher level of infestation, 2. Some people will even use a 3% level. In other words, 9 mites in a 300 bee sample is a 3% level infestation. So anything less than 6 would be under a 2% level of infestation. In the fall, once we've gone past the longest day of the year, towards the end of June, into July sometimes as well, we've got to go very serious of anything that we find in terms of mites. So 1% infestation or less, 1% or less infestation level. And of course, in winter, there's no brood. So the threshold concept does not work very well. But we still have a treatment thought, and that treatment thought might be, let's get our colony clean or with as few surviving varroa mites over the winter period. So we do have a treatment option in the winter of trying to clean the colony so that they come into spring, that we're going to have a much greater probability of getting that 1% infestation level. So that's our idea in terms of a threshold. It would be great if, in fact, this was valid. It is not. We're increasingly find out it is not. One aspect with this concept of this threshold, collecting 300 bees, we're making a basic assumption of the fact that the mites in that bee colony are randomly distributed. Okay, so the probability of a mite being on any bee is equal to being on any other bee. And so we're taking a sample of 300, and we have the probability of any one of the 300 bees being with a mite. Well, that is not true, as it turns out, because mites are clumped. For whatever reason, they are not randomly distributed. We don't know why they're not randomly distributed. So our basic premise of this being a accurate method of assessing the number of mites, a threshold, is not valid. Now, if you are sampling something that is not randomly distributed, there are other statistical methods of helping to make your sample then be a valid sample. So we do this all the time in biology. A distribution of something that occurs in forest patches that need a certain level of sunlight, for example. And if you went into the forest and you randomly sampled in the forest, you'd only find this particular plant, let's say you want to find, in patches that have sunlight. So that's not a random distribution in a forest, particularly in our forests of the Pacific Northwest. But we can still do sampling for that plant by then doing an analysis that is not assuming that random distribution. So we bias sampling towards, for example, patches that have sunlight, and we do it by levels of sunlight, for example, so we can do our sampling differently. So once we figure out why mites are not randomly distributed, we can get better with our threshold. So right now, we don't have that. We don't know that information. So right now, this is the best we have. Well, it's the best we have for example, fall, August through October. That's the best we have. But we're not there yet this time of the year. So what do we need to consider? I'm sorry, I want to back up 1 second with this slide. So our concept of a threshold, is a pest population will grow. We have a susceptible host to the varroa mite. So the varroa mites are going to grow. Any of the survivors are going to reproduce in the spring. The population is going to grow very slowly because not many mites are going to survive the winter. Not many honeybees survive the winter. But then just as our bee colony grows very rapidly, the surviving mites will reproduce and their population will grow rapidly. Now, the number that we're looking for in trying to do a threshold is a threshold. So that's this first line, an economic threshold. If we can determine that economic threshold, in other words, when the number of mites pass a certain number in the beehive, relative to, their proportion to the bees themselves, of course, and number of brood cells, of course, where they're reproducing. Remember, there's two phases of mites, both on the bees and in the brood. We then have a decision to make, and the decision in terms of an IPM strategy is, if we do some intervention at that point, at that particular point or prior to that point, can we then cause a drastic decline in the population of the pest? So the pest has to start all over the season again, okay? So now instead of 100 mites in a colony, can we knock it back down to the 2 or 3 that survived the winter colony? Because that's what was back here in spring, okay? In January and February, 2 or 3 mites, perhaps, maybe 10, maybe a dozen. But their numbers have now grown to hundreds. Can we knock that back down to a dozen? In other words, make that pest grow again. And of course, it's going to only have a few survivors because of our intervention. And so it's going to have that slow growth again. If we don't control the pest, is this other scenario going to happen, that they then pass a level where we have economic injury, EIL, economic injury level, so that those 10 mites became 100, became 1,000 mites, are going to grow to 5,000 mites. And at 5,000 mites, they're then enhancing the virus. Their DWV virus, for example, is growing now. And then we're going to have the chance of losing our host. They're killing the entire colony, or so debilitating the colony that we don't get honey from it, or they are so weakened they don't store enough honey, then they don't get through the winter. In other words, we need a cost benefit analysis here, so that if we do something, it's going to cost us time, cost us knowledge about mites. It's going to cost us some money if we use something that we've got to buy. What kind of benefit do we get from it? If we save a colony by intervening at this economic injury level, we have saved replacement costs, $250 for a replacement nuke. Or if you don't replace by nukes, you do splits divides, your time to make the split divide. The cost of a queen, for example, that you're going to put in your, $42 to put in your colony. So there are costs, and certainly time, your effort to make that split, however you do that replacement. So this is our concept of the 300 bees, that we're making an accurate assessment of those mite numbers are now 2 or about 2 past a level where if we don't do something, we can have an injury to the colony, to our pocketbook, to our host, the honeybee, non-survival or weakened colony. Now, the best of the strategies would be looking as well, not just at that threshold, but as the bees are approaching the threshold. So that's why we would suggest a May and April and May sampling. If you sample your colony in April and you find none, you sample it in May and you find 2, in the month that colony is doubled. The potential, if mites were randomly distributed, the number of mites in your colony just doubled in that month's period of time. So sequential sampling can help look at this valley, this period of time. Once it reaches this very steep level of growth, then you've got to sample more frequently. And once a month is not enough. If sampling 300 bees was very accurate, which unfortunately it is not. So it's gotten a little more complicated. Let me go on and talk about maybe there's something wrong with this county. They aren't producing anything useful. So we got all these different things to look at in terms of our county. And again, focusing on varroa mites is the one that is causing the greatest damage most of the time. So let me introduce what we should be thinking of now, this time of year. This is from the Honeybee Health Coalition. This is the Tools for Varroa Management, which I was a principal author, but we've had a lot of good eyes look at it and help with modifications as well. We're in our 8th revision, so make sure you're looking at the most recent one. But the concept that we want to work with is that bee colonies start from a low number, maybe 10,000 bees in a smallish county that survive winter in January, early February. They're going to grow slowly because there's not a lot of resources coming in. But then once we start getting to some resources, pollen, the first one, they're going to grow almost exponentially. So that's this pollination increase phase of our bee colony. And then they're going to reach a peak, so that 10,000 may grow to 50,000. So a 5-fold increase in the bee population. That's what we want. So that's what we want to happen in our bee colony. Now, we have to overlay that with the sweet spot. The sweet spot is this part in growth, and that's right now. That's this month for all of us. Starting for many of us starting in May, but certainly the month of June. Where the colony is now at a point where it can do more than just grow the family size. So when the queen lays eggs, she's populating her family, the bee colony itself. But the colony to reproduce then needs to rear a replacement queens and swarm. So the sweet spot is this part of the growth starting in April, certainly month of May and June for most of us, where that is now possible, where they are going to maximize the possibility of successfully raising a replacement queen and successfully dividing the colony with a half or more of them, the older adults, leaving, forming a bivouac and finding a new home site, and the original home site successfully requeening. So that's that part of their annual population growth. So that's what some of our management is focused on at the present time. But let me point out the other part of this equation. Not only are we managing a population of bees, because the mite is a vector of these viruses, we've got to manage its population. So the orange line here represents the population of mites. Now, I don't have numbers on here because there are a lot of variables, how much this peak is, for example, for both bees and in the orange case in the mites. But two things to point out. The mites peak beyond the peak of the bee themselves. So that indicates when bee population is decreasing, mites are still increasing and reaching their peak. The second thing is they are slower to get going in the spring because there are fewer that survive. Bees, once they can get pollen, simply for a while can outgrow their mite population. So bees will grow faster than will the mite population during the spring. But we're in that period of time now of this period. Now, I'm sorry. So this is the period that we've been looking at in terms of our mite intervention, the peak. But we need to do it sooner because at this point, as we have come to find out, it is very difficult to reduce a mite population that is high. Then it is easier, once we've developed a skill, to keep the mite population lower. We want to do this concept that we learned with the human population with the COVID virus, of course, the virus that produced the COVID symptoms, of flattening the growth curve. In this case, we want to flatten the mite growth curve. Back here, going back to this one. I'm not going to easily get back there. Okay. Back when I was here, this is where we want to flatten the bee growth curve. And we'll do that by taking frames out, by spreading brood out, by making 2 brood areas in one colony. In other words, our swarm intervention. So we do want to flatten the bee growth curve at this sweet spot at that particular point. But what we want to do in terms of thinking of our mite management is not at the peak. We're not worried about swarm control at population peak because they're collecting nectar. Where do we worry about swarm? As they're approaching that. Well, so too with mites, not at their peak population. That is August through October. Where we want to do our intervention is during this period of time in the growth phase. All right. Not when they reach peak, but in their growth phase. So alert, alert, alert. We need to control mites in the spring. A very bright graduate student at Cornell, I'm sorry, at the University of Maryland, who is not named Sammy Ramsey, as this is a fellow by the name of Zach Lamas, has discovered that mites are not randomly distributed. And secondly, he has discovered that mites are not where we think they are in the spring. If you want to hear him talk about this, you can look on you can look at the May and June American Bee Journal, May and June American Bee Journal, or to hear him talk about it is look on a website, the Sustained Beekeepers Guild, SBG, Sustained Beekeepers Guild of Michigan, MI, where he talks about what he has found with mites. Here from Bee Informed, importance of spring mite load. So here are 3 different resources you might take a look at. So he has found that in spring, mites are not in the workers, and they're not on the worker adults. They are in drone brood, and they are on the adult drones. So if you take a 300 sample of your bees in April, you're sampling workers, but that's not where the mites are. They're not on the workers, they're on the drones. To be effective, if that's an effective assessment of our threshold, we would want them to be randomly distributed and be on what we're sampling, the workers. Turns out, if his research is valid, and in research, it's not just what one person says, it's what is repeated by others, duplicating the results and with slightly different parameters, finding out the same thing. So just one person is not a definitive, this is the way it is. But in fact, now people are indeed confirming that in spring, the mites are not. The mites are not on workers and reproducing in worker brood. And this makes sense because their original host, the Asian honeybees, they don't reproduce on workers. The mites don't reproduce on workers, they reproduce on drones. So it seems logical that this mite, a transfer from those bees to the European honeybee, knows that. It's in its biology. If you reproduce on drones, you have a chance of reproducing yourself. If you reproduce 3 times over. If you reproduce on a worker, 1 time over. They're not dumb mites. They are a very successful pest. And so they know this, and so it makes a lot of sense. So, and as I say, some people are now coming up with some collaborative data that would indicate that this fellow is indeed correct. So if we consider that we don't have a way of measuring a threshold right now, then it's a little bit like our winter treatment to kill the vector, to get rid of the vector, the mite, that we've got to do some treatment without a threshold. So spring, now, still this month, what are some of our options? If you adopt that, in other words, we don't have a good way of assessing how many mites are in a colony. Sampling adult workers is futile because we're not getting a valid reading of what's in the colony. That the sticky board, the debris board is just too variable, that we've got to do some intervention, just like we might intervene in the winter period when there's not much brood. So what are our options in the spring? Well, we can have chemical, an IPM. This ought to be the last of our thoughts, but I'm going to switch those around and first talk about chemicals. In the spring, in our colony, we can use the impregnated plastic strip with amitraz. This product is Apivar. The issue is it's through 2 brood cycles, so it's a minimum of 6-week treatment, and then we should wait 2 more weeks to put a super on. So if you treated your colonies today, that means you would not be able to put a super on for 8 weeks. 8 weeks. Otherwise, you have the possibility of contaminating the honey that will be stored in the colony. Then harming yourself, harming the customers that you're going to sell the honey to, and your wax becoming a sink for this chemical, because the chemical is lipophilic. In other words, it's absorbed into wax. So that's not usually a good option. It is if you start in January. And so if you've got anyone along the coast that winters their colonies along the coast and then takes them to almonds, they're going to put Apivar in their colonies on any winter day they can. And any day in January they can get in their colonies, they're going to put these strips in every one of their colonies, because then they've got that 2-month window. Now available is a new product, concentrated of the amitraz. It's going to be labeled as Amaflex. It's from the same people that make that sampling jar. It's put on as a gel with a device to add the gel. It's put on just a simple strip of luminized paper. It's a flash treatment. It doesn't need to be for a 2-month period of time. The problem right now is it's not available. They've had trouble with the manufacturer. It's manufactured in Europe. They've had trouble with the manufacturer, and so we will not generally be able to get it until the end of the year. So this might be a tool for next year, but it's not a tool for this year. So Apivar, the one tool we have, the synthetic, not a good option. We don't have enough time. So what are some of our other chemicals? Formic. Formic, we can use a half dose legally. So instead of 2 pads, we only put 1 pad on. So this is formic acid soaked into a absorbent pad. We put it on top of the frames. The problem is formic has a lot of collateral damage. It often will, we need to try to avoid the queen or there being, killing the queen, or there being egg laying damage. So usually, often when we use formic, the egg stage just completely disappears, and they empty cells out of the young larvae, for example, and often the queen just stops laying eggs. She's still capable of doing so, but for whatever reason, because of the chemical in the colony, she stops laying eggs, and this can be for up to a week's period of time. And remember, we don't want to normally in the spring break that curve. Now, as a swarm control technique, we can also use this as well in terms of swarm control, because now this time of year, if we kill the queen, so what? We're still going to get our honey harvest. You don't need brood to get a honey harvest. You need bees, adult bees, to get out to the flower. So this is an option, particularly now in this month, as a way of trying to do a treatment. The third option in the chemical realm is Apiguard or Apilife-Var. Here's the colony with a spacer rim on the top, and that's the gel, the Apiguard gel. This is a thyme oil. This is an essential oil. We use thyme leaves. Some of us use them in our spaghetti sauce, but we only put a little bit in. Some people use thyme oil as a treatment for some types of cancer, digestive issues, for example. But now we're talking of concentrating the essential oil that it's a poison, that it's a killer. And so it can cause harm to the bees, cause harm to us as well. So we have to be careful with its use with all of these chemicals. But the nice thing is that it will work this time of year. The issue is if you are ready or you have supers on, this is not your treatment of choice, simply because it will impart a potential taste to ripening honey. And it's a Listerine type medicinal type of taste to the ripening honey. Once honey is fully ripened, it's not a problem. It apparently doesn't bother the bees. But if you have supers on, this is not going to work. So we're at the point with many of us that as a part of our swarm control, we've got supers on county. You got to get that incoming nectar out of the brood area. You can't let the bees backfill the brood area right now. If you do, you're likely to have swarming if the main nectar flow is delayed. So let's get to the IPM part. So what can we do? If in fact the student is correct that the mites are in the drones, let's manage drones. Let's manage drones like the Asian honeybees manage drones. So let's put the drones where we can find them, where we can remove them at the capping stage before the drones have a chance to come out as adult drones, and with it, the foundress mite and her three mature daughters. So what we do is we raise drones to the drone capping stage, those dome-type cells, and then we murder the drones. We take the drone brood out of the colony. If we use this green frame, the green colored frame, that's drone foundation. So when the bees draw the cells, they're drone-sized cells. The queen comes and lays the unfertilized eggs. And then in this case, a full frame of capped cells. And we've removed them. We put them in a freezer, kill the drones that are developing inside overnight, and then take a capping scratcher and remove the cappings. Then we're going to snap the adult drones, the larval, the pupil drones out of the frame, snap them out, and then give the mess back to the bees to clean up, repolish the cells so that the queen come back to lay eggs. This works if you manage the drones. If you've got combs that have drones here, there, and everywhere else, this doesn't work. They often completely ignore this green frame. You put the green frame on the edge of the brood area, not in the middle. So there's management. There's a technique on how to manage this more successfully than in others. But if the bees are rearing drones wherever they want to rear drones, you are not managing them. The bees are managing you. And this drone brood removal with this green frame does not work as well or as efficiently. Okay. So that would be our first choice. In other words, the non-chemical means, if we're following an IPM principle in terms of our strategy. Now, I like to equate IPM as something along this line. For your projects that you have to do around the house, around the ranch, in the barn over the weekend, you can take your toolbox and put a tool in the toolbox, but are you going to be able to do all the various types of things that needs to be done? So if you're going to go with your toolbox to get varroa mites, does your toolbox look like that, or does your toolbox look something a little bit like this? All right. The other aspect is you can put a nail into a piece of wood with your fist. You can use the back of the handle of the screwdriver. You can use a pair of pliers, or you can use a hammer. Well, most of us would want the tool to put a nail in a piece of wood to be that hammer. What tool are we going to use to get rid of, to help our bees with varroa mites? Not get rid of them, but help control the mites, control the population in the spring. Are you going to use a tool that doesn't work very well? Or are you going to use a tool that works like the hammer to put that nail in the wood? I think most of us would opt for, let's go for the hammer. And so what's our hammer in the spring? Our hammer in the spring is drone brood removal, if the student is correct. Not one of the chemicals, not the back of the screwdriver, not our fist, but a hammer. Okay, drone brood removal. Just like in the winter, when we would not want to make a sample of bees, what's our best tool? And in the winter, our best tool is oxalic acid. That's the very best tool we can use. Not the other chemicals. There's no drone brood to remove in the winter. We don't want to divide a colony during the winter. So what's our best hammer to hammer the mites in winter? It is oxalic acid. So does your toolbox for combating mites, helping your bees combat mites, does it look like this toolbox? Are you trying to use all the tools that are possible in the toolbox to combat the mites? All righty, I've gone much too long. Let me move ahead to a little bit. I mentioned this triangle in terms of our pest management. let me mention just a couple of things that we can do that are not expensive, that are preventive more than interventive, that do not involve some level of toxicity. What we can roughly label as cultural. We can improve the stock, have more hygienic bees, for example. Bees that are better able to fight the mites themselves. We can't do the whole job for our bees. The bees got to do it itself. Keep our colony small. Some people like to do this as fewer manipulations, but we can keep our colonies growing out of the mites, okay? Spread colonies out. Give our colonies a distinctive address. Each of these colonies is a distinctive address for the bees that are foraging. Get less drifting, the possibility of the mites being on the back of bees going horizontally. We can capture swarms from bee trees. Bee trees, by definition, are successful, not beekeeper swarms, but bee trees if we're in an area, because those are colonies that survived. Not many colonies survive bee trees, okay? Less than 20% that go into a bee tree actually survive that first winter. But if you can capture those swarms, they are survivors. They were ahead of the mites. And so when you put them in your hives, they're going to be ahead of the mites. And with that as well, go ahead and euthanize. If there are too many mites, don't leave that spreader of mites, potentially from robbing, from drifting, in among all these other colonies. Get rid of that one particular colony. Euthanize if there are too many mites, if looking at in terms of mites. Feeding bees makes them healthier, whether you feed with a liquid syrup inside hive feeder, for example, feeding them protein, and when it's real cool, feeding protein, letting them go fly into the dry pollen, for example, or feeding them the protein inside the colony itself. Now, there are mechanical devices that we can also incorporate here. Mechanical are things such as doing something that is going to cost you time, maybe not a lot of money, but time. So do the brood break, allow the bees to swarm, or if you prefer to manage your bees rather than having your bees to manage you, you do the swarming. And in that we call splitting or dividing colonies. In other words, before the bees have a chance to split themselves, you do it. And you raise queens or you purchase queens. You create an artificial brood break and you push in a queen cage with a new queen. The powdered sugar shake, some like that because it's somewhat benign as well as screen boards. So you have a little bit of investment, a little bit of money in the powdered sugar, a little bit in a screen bottom board, but this is helping to rid some of the growing mite populations. But I would suggest that with our newest finding, particularly of the student that as we validate that he's in fact correct, that using the special drone foundation to go ahead with drone brood removal. So here with our brood break, we're going to confine the queen. This is a push-in cage, one you can buy. The bees can feed her through the screen on the top. You push this into comb, so she's on a little piece of comb. You might do the brood break by confining a whole frame and putting the queen in there. And then you can get rid of the brood and treat the rest of the colony, take her out while the treatment is going on, for example, with formic, so you don't kill her. So there's a number of ways. Again, these are traps. This is a mechanical device of some way. Here are the green frames. And again, the proper placement and getting rid of the drone brood. Now, there's another technique if you don't want to put out the money for a green frame, is if you have full-size boxes, put a medium frame in. So the medium frame is going to come up down to there's about a 2 to 3 inch gap underneath the bottom board. Just a whole gap. The bees aren't going to like that gap. And this time of year, if you are managing them and controlling where they have drones, the replacement comb they put in there is going to be drone-size cells. So you have worker comb in a medium frame in a full-size box, and the bees build the drone comb where it is. You put a thumbtack on that frame, and then you remove them at the capping stage. And all you do is run your hive tool along that bottom bar and scrape off the capped drone brood. Now, if you're going to use this technique, any trap, it's effective when you use the trap effectively, and ineffectively as you let the drones hatch. Okay, let them come out as adults. He closes as the adult stage, because when they do so, they're going to be you're then generating mites. You now have a mite generating colony, and you're not controlling them. They're controlling you. And then for distribution of mites in colonies that are doing a good job, holding back the number of mites, the bees themselves, in the fall, putting on robbing screens so that you keep out both the predation from yellow jackets, but in addition, keep out the robber bees that are going to bring mites with them. Here's that green frame. This fellow puts them in the freezer. So he has 2 in a freezer now. He's taking the capping scratcher. He's removing the cappings after it's been in the freezer about 24 hours. And notice the placement. So here's 1, 2. And so this is the second. So here's frame 1, a fully expanded colony. This is where that is going to go right here, the second frame, because that's going to be on the edge of where the cluster is. Don't plop these right down in the middle. And this is obvious that get rid of those. How many of these cells have mites in them? And the chances now in spring are a good number may have mites in them, being very successful in their reproduction. Okay. Climbing up our ladder, now we're getting more than prevention. We're doing more towards intervention. Biological, the only one that seems to have any promise are certain types of mushrooms, myceliums of certain types of the shelf mushrooms. There's a company working on developing this that we could feed this in sugar syrup. It's not there yet, but it's being worked on. So we effectively don't have a ladybird beetle, the ladybug that will attack mites. The synthetic chemicals, I've talked about those. These are the things that are not as toxic to the environment and to us as the human applicators. So these are the essential oils, the Apagard, Apalife Var. Again, this is the Apagard. Essentially used the same way. And then the formic acid. And finally, this group of, that one's out of place. So this is also the acids, the formic acid, oxalic acid, and the Hopgard, the 3 different acids. Of course, we have different ways of using them. Here's the full treatment of formic. This is a fair chance of killing your queen or stopping her from egg laying and killing any open brood when you use it at the 2-pad treatment. 1 pad will have less of an impact. You end up treating with 2 pads, but you sequentially treat. You put 1 pad on, wait, and then put the second pad on rather than the 2 pads at one time. So there's, again, management of how we might use formic acid. And then oxalic acid, anytime there is brood in a colony, this is like trying to put that nail in with your fist. It's not an effective treatment. We're trying to work to make it a more effective treatment. So our approved methods are dribble, where you mix it in sugar syrup and put it in with a syringe, and fumigation, where you change it from the crystal form to the gaseous form. This only works effectively when there is no or very little brood, but you can manage and get a brood break. So there's very little brood, and you can use these materials. Of course, Randy Oliver is working almost exclusively alone and trying to find a way to put this material, oxalic, in as an extended treatment, absorbing it into something, glycerin or canola oil, and then putting it on pads, something like this, as an extended treatment. It's not approved. 3 states in the East try to do a state improvement, but EPA did not allow it, where they could have a state exemption. And at current levels, current way of thinking, EPA does not want to give approval for homemade remedies. There is an opening for a company to come up with this technology. Well, one has, a company in Argentina, but they're not going to ask for US registration. So it will have to be another startup company. So if you've got a lot of money to give to a startup company, and it's going to take a lot of money to get it through EPA approval and 2 to 3 years, maybe in the future we could have this. This does seem to have some possibilities of working in the West, where it's drier, much more easily than in the more humid eastern states conditions, but it does seem to have some possibilities. And then the material Hopgard, the nice thing with the acids is you can use any of them, formic or Hopgard, particularly when supers are on a colony. So you do not have to take supers off to go ahead and do your treatment. Formic is a chemical that is an acid that is in honey. So by adding formic in these pads, you're not increasing dramatically the amount of formic acid in honey. And so it's not harmful to the product that we're harvesting. Of course, it's tough on the bees, but it's not harmful to us or the treatment. And then the synthetics, that meant to be the top of that triangle. This is the dog collar approach, impregnated plastic strip. We don't put it around the neck of every bee, but we put it in by strips in the colony. One strip for every 5 frames that there is brood. So only 1 strip in a nuke, or perhaps a half of a strip in weak colonies. And the problem is you've got to go for effectiveness, the 99% effective. This is the gold standard. 99% effective. It has to go through 2 brood cycles. So it's got to be in 6 weeks. That's certainly the downside. The other downside of this material is this is lipophilic, and so it's absorbed into the wax. So if you have used it at any point in the last 100 years, it's been on the market about 30 years, and we take a sample of your wax, we're going to find it in the wax, not necessarily in the bees or not in the honey, but we're going to find it in the wax. And of course, then if bees put the honey in that wax, it can then be absorbed back out of the wax into the honey. So all of these treatments have a negative aspect to it. I mentioned this resource, tool guides. You can download the guide. Make sure you get this last edition. That's what's now up on the website. Tools for Varroa Management from our Honeybee Health Coalition. Fully downloadable, no cost. We have the videos that go with it. They're embedded in the download. So if you've never used the synthetic amitraz, for example, there is a instruction sheet in terms of how to use it. If you've not used the hop guard, for example, we have that, the acids, etc., as well as some others about mites and the importance of an IPM approach. Tools for Varroa Management. Finally, prevention is better than cure. Monitor mites frequently. That's been our mantra, but it's going to have to change. What we actually are going to need to do is tell you how to monitor drones, how to look into drones in spring, so that you do have an approximation of what kind of number of mites are there. And if you then do something such as drone brood removal, are you effective? Right now, if you use drone brood removal, how do you know that you are flattening the mite growth curve? Okay, there's no way we can tell you, but if they're in drones and we can do more effectively sampling of drones, then we can get that intervention well. And then with monitoring, intervene early, 1%, 3 mites, no more than 3 mites per 100 worker bees. Then that it may well be in April and May, a raging infestation, because most of the mites are in drones. Learn to recognize healthy hives. It starts all with that, both brood and adult individuals. Can you look a bee between its eyes and tell if it's sick? Okay. For those of us have gone through kids, one of our offspring at some point, grandkid comes in and says, "Yeah, I don't feel well. I don't think I should go to school today." So what do? You kind of make that bead between the eyes. Are you really sick? Are you trying to fool me? Well, look your bees between the eyes, and are you sick? Are you healthy? What might be the case? Of course, in bees, it's more of a challenge. In each eye, they've got 3,200 facets. In other words, eyes, 3,200 eyes on each side. So it's a little bit tougher to figure out which ones to look at, right? Replace older frames and combs because of these inoculums. They're there. With dead outs, thankfully, the mites do not survive. However, we are finding that some of the viruses may survive in from dead outs. That old moldy stuff, the only way we can do it is give it back to bees. Cull out the older stuff. Don't rotate out your old equipment or cull frames. Be very wary of used equipment, especially used comb where there still is comb itself. If you do not know how to look for scale of AFB, Don't ever, ever accept someone else's comb. Don't accept someone else's colony as a gift colony. Don't let a good, friendly neighbor move their colony into your apiary. Because if it has American foul brood, there's a chance that your colony will get those spores. Your colony has healthy brood, and the spores will grow in your brood. And finally, with all of this, these are pests, and in your club, you've got some good beekeepers that have unfortunately had the occasion to see some of this more than once. We don't see this enough. We really don't. Thank goodness we don't see it enough. And when we do, we got this, well, is this European foul brood, or is perhaps this is sac brood? No, maybe this could be American foul brood. Well, what you need to do is get a sample, go to the expert, do the test with the Ropey test, the Holtz milk test. Ask someone that's had in the club, that's had some experience. Take out the frame where you think it is suspect and bring that carefully to a meeting the next time, or to that expert, and ask them, or send the sample into a lab that is certified. The U.S. Department of Agriculture lab, the bee lab in Beltsville, Maryland, will do that analysis for you free, no charge. So ask for help. We can't be expected, with all the other things we're learning about bees, with swarming, with when to super, when to add another super, where to place that super. Is it time to harvest? Is this colony rearing enough fall bees to get through the winter? All those other questions we've got to ask. Asking about these pests and what might be effective treatments is a lot more. So ask for help. Don't be afraid to ask for help. You've got experts there locally that can help you do that. And finally, in the shameless promotion department, some books from me. So I'm going to stop finally and see if you've got some questions. And I'll go to a look at some of you. My
Wow,
God, we still
that
got
was
25 survivors on the.
I'm gonna have to watch this again. Has a lot of info. You want to
Thank you, Eric.
go ahead and read the chat, or do you want me to? You got some
What we
questions
got
in
in
there.
chat? Yeah, let's go look at chat. The tech and RMPs has been okay. Here's the presentation. Here's the presentation file. Glad the tropicale mite is not on the list yet. That is being looked for in some sampling, including in the state of California. U.S. Department of Agriculture, APHIS, APHIS. APHIS is the agency that's charged with it. They do sample in better than 30 of the states, finally, and part of their sampling looks at for the possibility of that introduction. Do you recommend using new foundation every year? No, I don't. Drawn comb is our most valuable resource. What I do recommend is replacing, after 3 or 4 or 5 years, replacing the older aged comb, the darkened comb, with foundation. So I recommend each year trying to put in 2 or 3 after the first couple of years. You survive the first couple, 3 years. Thinking about replacing your old drawn comb then with new drawn comb. But not replacing every year. That you're the manager. You certainly have that option, but there's a cost to the bees drawing the comb. So this could be a good management strategy of replacing 2 or 3 every year after you've had several years in with it. Related to euthanizing for too many mites, do you recommend all destroying bee bread from those high mite colonies, or can it be safely given to another colony to use? What we're going to euthanize, of course, is the adult population, and then any of the brood that is capped can also be killed. Unfortunately, the best euthanizing agent we have is soapy water. The wax cappings are permeable, so if you kill all of the adults with soapy water, you're going to kill most of the brood as well. Because it's going to be sitting in that soapy water, and so that's going to kill the mites. So certainly the honey frames, any of that can be washed off afterwards. The bee bread as well can be washed with clean water to get rid of any of the soapy water residue. and give that to another colony.
I had a question about that one. That was my question.
Okay.
I was wondering along the lines of, you talked about the viruses being transmitted through the comb possibly. Could viruses also be transmitted through the bee bread, or is that unlikely?
Unlikely because of the way method of its processing. It's anaerobically processed. So viruses are pretty sturdy things, but viruses need living material. So not likely. The viruses we have found on stored comb, for example, from a dead out, are not the serious APVV or DWV, not deformed wing or acute bee paralysis. There's some of the others. So generally with a dead out, we say, give it back to the bees and let them clean up the mess. Now, if you've got Africanized bees and you need to euthanize a colony, in other words, a very defensive colony, and not just one-time defensive, but they meet you at the apiary gate, they follow you, they persist, you get hundreds of them bouncing on your veil as you inspect the colony, and you need to euthanize those colonies. You need to euthanize adults and brood in that case, both adults and brood. Because the new brood, what has happened is that queen is mated with a couple of drones that are Africanized drones wherever the queens were mated. And so with a very small population of Africanized bees, the colony can become very defensive very quickly. So if you need to euthanize it, you need to do both adults and brood. In the case of mites, I'm sorry, yeah, in the case of mites, if you separate the adults, then don't allow that brood to hatch and have the reproducing bees then beyond the new adults that come out. So you can separate the cap brood from the open brood, or just get rid of the brood as well. Does that help, Angela? Any further on that?
Yeah, it does. That helps a lot. Thank you.
Oh, okay, good. How long do you recommend freezing drone brood to ensure the mites are killed? Good freezer with everything else frozen. You're just putting in a couple of frames. A day will work. As I said, one person will put 2 in and then take 2 that have been in the freezer. You need to get the bees to draw the drone comb, and then you can replace the drawn comb if you don't destroy the drone cells about every 3 weeks. Okay. And so he'll leave the drone. He has more than 2, so per colony. So he'll leave the frames in for that duration until he's ready to remove another green frame. But if you do it and you only have 2 per each colony, you only buy that number, leave them in a day, then take them out, do the uncapping so that and then shake out as many of the drone pupae as you can, and you won't get all of them. And then let the bees clean up the mess. The good advantage of that is that the cells, the drone cells are mostly still intact. If you use a half depth frame, you're going to cut out the whole frame, the whole half depth part, the lower below the half depth. You're going to cut it out. There's no way really to salvage it. So they got to build it back, but they'll build it back real quickly in the spring. So this only works roughly May and June into July. Is this a F- question? Oh gosh, should we be sampling drones in spring rather than workers? I wish I could give you a definitive answer. I think the answer is going to be yes, but it hasn't been worked out. That's not an F answer. It's a fantastic question. F- question. It's a fantastic one. The problem is, so as a rule of thumb, you can look on adult drones or you can look on drone brood. So if you use a capping scratcher and go into your drone brood, wherever you find it, between the boxes, in the bottom of frames, in the corner of frames, wherever, and you decapitate drones that are fully formed in the pupal stage, you can quickly look and see how many mites. So if you find mites on drones, and I would suggest that you're going to have to sample somewhere more than a few. So let's just give you a round figure of 75. You're gonna sample 75 drones, and if you're finding that you are finding mites, an adult mite, that founder's mite, On more than a couple of those drones, then that's intervention time. Okay. And you've just intervened, of course, by getting rid of those drones. You've started the process of getting rid of some of them. So then you either go to your chemical or you continue with your drone intervention. Now, that's a ballpark estimate. If you find them on 75 and you find them on 5 drones in those 75, you are going to probably lose that colony at the end of the year. I mean, that's that serious. That's how quickly they're going to reproduce. So a couple, maybe not intervention, but certainly 5, I think so. But that's not a threshold number yet. We're trying to come up with a better threshold than that. And in terms of adult drones, pick them off, pick them off, pick them off. Or bring your favorite chicken with you so the chicken is picking them off as you're looking at your bees right off the frame. Train your chicken to pick out the drones, not the worker bees. How you do that, I don't know. But sorry, I missed the beginning of the meeting. If anyone can point me to any local queen bee sources available currently. Eric, any help? Someone can help someone on that one? Someone's asking
Yeah,
if they
that's
can point to local queen bee sources locally available currently.
yeah, it's kind of all over the place. We've been getting a few inquiries on people looking for local queens, but you know, it's a little dicey up here. Yeah, I don't have an answer.
Also has been quote a local source over in the Rogue Valley of Oregon. Another source is Dig and Livin' down below Cave Junction, right on almost on the border with Northern California, off of 99 there. There's a source, a fellow up into Oregon along the coast. I can't think of his name. Fellow by name of Noah in the Appleton Valley, Cave Junction in Oregon. So those are some
Honeybee
of the
genetics.
epigenetics.
Yeah, honeybee genetics.
Yes. Yeah.
I have a question. Can you hear me?
Yeah, I can hear you. Go ahead, Rick.
Okay.
Seth.
I have several questions for you. One, the first part of the question is, what do you think of Dr. Wolfgang Ritter's work that he developed several years back about in the middle of the honey flow, removing all brood except one undeveloped frame for 1 week as a bait frame, and then extracting that a week later and replacing all the combs with foundation frames?
If the flow continues, that will work. Replacing with foundation is at the expense of a potentially greater harvest. So yes, that works. It's a big timing issue, and where you are, it needs a more of an extended nectar flow. I don't know what the flow is where you have your bees. If it's a short, intense flow, that system is going to be a tough one to get to work management wise for you.
Well, I'm
flows.
wondering do you know of anybody in North America trying these approaches in a concerted effort to try to bring, I see it more as maybe making a divide, and the divide will be the future. And the hive that was the mother hive that you pull the brood out of will be the hive that produces maybe less honey, but that's the hive that's expendable, where then you put your energy in the new divide for the hive that will winter.
Yeah, there's a lot of people doing that. So they're making their nukes up and getting new queens into nukes, whether they're rearing the queens themselves, buying queen cells. If you've got a local producer that's going to be selling the queen cells rather than adult queens, or buying queens in. And then next spring, you have colonies that have been started as colonies in the fall, starting initially as nukes, and then either overwintered as nukes or overwintered as full-size colonies. In other words, the nuke is a resource, and then we reunite the resource back to the colony after killing the colony queen. that's going to help you an awful lot at that sweet spot. In other words, right now with swarming. So if you have a real serious problem with swarming every year in your bees, that making up nukes in the fall, requeening in the fall is a marvelous, marvelous management because new queens that haven't been through a spring are much less likely to start the queen cells to start swarming. So that management is fairly common among North American beekeepers. The aspects of replacing all the comb, moving all the brood to one colony, most of us will do that a little bit earlier. A great time to do that brood break is right before the flow. A better time is, I would say, is remove the brood at that point. You still have the adult bees there, and then you can treat the adult bees with oxalic acid because now you no longer have that brood where all the mites are. Oxalic acid is putting the hammer to the nail in that case. In other words, you're hammering those mites that are on the adults. The problem is if you got 2 or 3 colonies, many of these techniques don't work because what do you do with all the frames of brood? What do you do with all the new colonies you've created? This issue of colony creep. But from that aspect, there are a lot of people doing that. Yes.
Okay, the other part of the question is, I was talking with some Hungarians on the web, and they actually build little queen excluder cages that they fasten to a frame, and they actually will cage the queen in the late summer, early fall for 24 days. And they're being successful with that. Do you know much about this?
Yeah, see, if you Google the word queen sequestration, you might find more information on that. Generally, they're building a queen excluder cage around a frame with a trap door, so you can put the queen in there. And some of them build a cage so that you can remove. You can actually buy this as well. This is available. A wire cage is better than queen excluder material, by the way. Just got to be careful you don't damage it. And then when the queen has pretty much used that frame for all of her, filled it up with eggs, you can actually pull that frame out. You know the queen's there. You find her, take her off with lifting her off with a queen catcher, and put it on a brand new frame in that cage. So you can actually manage that, and then what you then can do is remove the drawn brood, the cat brood, and do your treatment with oxalic acid. Oxalic acid is not hard on the queen. So yes, more and more people, I understand, more and more people are doing that as a management, sequestering the queen in a cage on a frame where she can lay eggs. And managing the brood that then, is the result from that, and in the whole colony. And why 24 days is that creates a brood break. Actually, you don't have to go 24 days. If you do the math, you'll find out you can do this starting at 12 days. And then in the original colony, most of the brood will have hatched, and you can use oxalic acid in your colony. The closer you go to 19 days, you will definitely have a 2-day window. You don't have to go 24 days. But in terms of getting the queen back in the colony, 19 and 24 days doesn't seem to make much of a difference. There's always risk of losing the queen.
The method that I saw the Hungarians using, they didn't keep the queen on a comb. They actually kept her in her little 4-inch by 4-inch box of queen excluder. And so the bees could still freely get to her. And years ago, more than 10 years ago, I caged up 80 queens and into queen cages, and I had to reintroduce each queen to their colony because the bees couldn't. Are you still there?
Mm-hmm. I'm trying to find the one I showed you.
So I was interested in this method of having having a serious brood break with no brood whatsoever, having the queen available to the workers, and it seems like they're having really good results, and they live in an environment that is much less forgiving than what we do on the Pacific Northwest coast.
Yeah, for sure. Sure. So I went back to this slide. So this illustration up above is exactly what you're talking about. So it has spikes here to push into frame itself, so that you're putting this on some comb cells, and then this is so that the bees can actually feed the queen. So in this case, you're caging the queen by herself on a frame, but the bees can take care of her during all of that time. So this is a commercially available push and cage.
I have one of those. That's very different than what I'm referring to, actually. That what I'm referring to, what they use in Hungary, is they make a little wooden frame hanging on an empty frame with queen excluder on both sides of the wood, and there's no comb. That's how they're doing it in Hungary, and they've been doing it, and they have good survival rates in the winter from that method.
You're caging about 20 cells here, so I don't see the difference between a wood cage versus this type of a cage. But okay, I'll grant you that it is different. Yep. Okay. Whatever rocks the boat. Yeah. You're right. It's very, very tough environment that they're talking about. What most people now seem to be trying is to actually cage a queen in a whole frame and have the ability to take that whole cage out, take the frame out of the cage, take the queen off that temporarily, take that frame out and substitute another frame. Now, the difference with caging her in a cage where she's not going to have any egg laying, whether it's the California shipping cage, whether it's the 3-hole Benton cage, caging the queen, or in this case on wood itself, with excluder material around it, is that queen is going to be degraded. In other words, she is aging. Yeah. She's not laying eggs. And so is she going to bounce right back after that period of 24 days of sequestration under those conditions?
Right.
Versus sequestration where she's on a frame with drawn cells. I can't give you the answer. I haven't done the work, but there may not be a difference. Great. Anything,
well, is there
anything else? Go ahead, Seth.
I'm just wondering, is there any groups doing science behind these methods with data that's quantifiable, which can be a cultural practice where we can actually get away from putting things into the beehive? There should
can't
be.
point. There should be. It absolutely should be. I can't point you to. I can't, for example, drone brood removal. I can't point you to. Gene Robinson did some work at University of Illinois. I can't. A fellow that in Saudi Arabia has done some very good work on this, by the way, as well, on drone drone brood removal. So there's a little bit of work on drone brood removal. On this new stuff of the brood break, no. Other than a break where they just simply remove the queen and then requeened it after a period of time. There's not much data. No. Ellen Topitzhofer has, with Project Apis M on its website, has a very good free publication on queen banking. And banking a queen, we're trying to take a queen and put her in a cage and keep her alive and in good shape for a period of time, 6 months. And then have her be productive when we then reintroduce her to a colony. So there is so she goes through the science in terms of queen banking. So that would be that, but that's almost all that data is putting her in a small cage, which would not be different from what Seth is talking about with the Hungarian people doing with a cage in a wooden box.
There is
most of the cage, all the cage work that I can think of is is the queen herself, maybe with some attendants, maybe not, but not sequestered on comb. I can't think of it. There may be some literature out there, but none that I know of off the top of my head. I'm sorry, that's not a very good
Well,
answer.
I appreciate your talk very much.
Thank you. Thank you. Catherine, it's too late to install green frames. We're coming upon it. After the end of June, it doesn't work, but it may be too late for you this year because you have drones everywhere. If you can go in and rogue out all the other drones everywhere now in your colony, the green frame will work spectacularly. In fact, the green frame, bees drawing the green, bees don't like that green frame. Even when you add more, and you should add more wax to it when you put it in, melt some wax on it to make it more attractive. But the bees are sometimes very reluctant to draw out those green frames. So you've got to manage the bees. You've got to get rid of virtually all the other places right now in the colony that are raising drones and make that green frame the only place. And of course, they've got to draw off. So this time of year, you're only going to get about one more cycle out of it. In other words, on one side of a green frame, we're looking at about somewhere between 2,500 to 2,700 drone cells. Those couple of frames that I showed you, remember those frames I showed you? Let me go back to, again, no, here we go. I'm going back and finding that one green frame that I showed you. In the talk. By the way, there's going to be a quiz on this at the end. Did I not tell you that? Where in the world was that green frame? I hope I'm not making you all too dizzy here. Here we go. Here it is. So look at that green frame. I mean, that's a full frame. I mean, they've worked beautifully. And in this case, actually, you can see the green frame is next to the internal hive feeder. So here's the edge of the box. Here's the feeder, and that's where the green frame is. Everyone see that? Yeah.
Yeah.
Yeah. That's
All right,
working spectacularly.
We got one more question in the chat.
Okay, what's that one? So it's too late. Okay, we got Catherine.
You just answered it, actually.
Okay.
You said remove.
Oh, Mary, yeah. Okay,
Get rid of all the other places raising drones.
Yeah.
For
Yeah.
next year.
They know where they're rearing drones, and they've got a head start on you this year, next year. Eric has a product, green sequestrin. Oh, great. Thank you, Eric. So here's a website. Green the queen sequestration. That was the kind of the word you'd use in your search engine. So that's how you found it, Eric. Queen sequestration.
Yep.
Nice.
Yeah, it looks like a fun thing to build.
Yeah. This time of year, bees will build drone brood readily in foundationless. Oh, okay. Yes, of course. Janine, excellent thought too. The third option, I mentioned green frames or a shallow frame, medium frame, and a full depth box. The third is simply the frame itself. Put the frame itself in the colony. The middle of the frame to anchor it may be worker brood, but then the wings of that frame, very frequently, they'll go and build drone cells. And Mary, this might still work for a colony this time of year without much of the drone roguing. So then what you do is let the queen come in, lay eggs, and then all you're doing is cutting sort of so you got your frame, you're cutting off the wings of it where the drone, and we can tell drone cells from worker cells. So you're cutting off, once they get to cap stage, you're cutting out those drones on that wings of just a simple frame, no foundation. Yeah, thank you. That works well as well. So that's sort of the third way to you manage drones versus drones everywhere here and there is the colony managing you. Isn't that a nice thought? You know, we are the beekeeper, we are in charge, we're managing our bees versus what's really the truth. The bees are managing us. So these are a couple ways to take back the power.
Thank you so much. That was a wonderful presentation.
I hope it wasn't too general because as you were starting, some of you people had some very specific questions in terms of disease. Eric, you might say you were mentioning that some people are seeing a little bit more of this shiny black hairless syndrome, what we think is the acute bee paralysis virus.
Yep. It looks like Angela Stewart, I think you covered that. I mean, I've never seen it to the extent that she's seeing it.
Yeah, I just found it today. It was quite a few bees. There's a big old handful of the black shiny bees outside of a mating nuke. And I've seen it before. I've seen 1 or 2 bees kind of crawling around and I just pick them out, throw them at the chickens. But this one was pretty, it was notable, definitely significant.
If you're starting to see them outside, crawling up on those grass blades, seemingly unable to take off, you know, that type of thing, you're seeing like the proverbial tip of the iceberg. There's still a, you know, that virus is getting a head start on the bees. And immediately knocking back the mites might work. So a drastic, the formic acid, get rid of as many mites, the flash treatment might be the way to go, even though you might lose your queen. You're going to, you know, lose maybe queen egg laying, but you've got to get a fast flash treatment in there. And of all the fastest of the treatments, that's it. That's the formic acid. Because if you're seeing that much, it may just be a ghost in the fall. In other words, they store you honey and it looks good when you look at them in August, and then in October, there's nothing left. Bees are robbing, stealing, you know, are robbing the nectar. They're coming in and stealing the honey, the capped honey. And it's a dead out by November.
Okay. Wonderful. Thank you so much. I'm going to stop
Alrighty,
recording.
I've gone over my time. Thank you, Eric. Thank you all. Appreciate it. Good luck with bees the remainder of the
you.
season. Thank you.
Yeah, thank
I
you.
look forward
Thank
to
you.
your next visit. Thank you very much.
Thank you very much.
Alrighty. Good luck with your bees.
Thank you.